Good evening. I'm Hank Levy, and I'm director of the Paul G. Allen School of Computer Science and Engineering. And I'd like to thank you for joining us to the first presentation of the 2017 Engineering Lecture Series, Borrowing from Nature to Build Better Computers, DNA, Data Storage, and Beyond. This series is presented in partnership with the College of Engineering and the UW Alumni Association. If you're not a member, I encourage you to visit the Alumni Association at uwalum.com and learn more about the benefits of membership. If you have a big collection of digital photos or videos or films or other files, you'll appreciate tonight's lecture. In today's world, using our phones and computers and numerous other devices, we are creating and saving astronomical amounts of data. As just one example, YouTube said several years ago that users are uploading 300 hours of video every minute. That requires that they buy thousands or tens of thousands of multi-terabyte disk drives a day. Now imagine, fast forward a couple years, when all those videos are much, much higher resolution 3D virtual reality videos. What will happen? At UW and Microsoft, computer scientists have been working together to develop the next generation of data storage inspired by nature. Tonight's speaker is Professor Luis Ceze from the Allen School. Luis grew up in Brazil, where he received his BS and MS in electrical engineering from the University of Sao Paulo Polytechnic School in Brazil. He completed his PhD in computer science at the University of Illinois in 2007, and we were extremely lucky to have Luis join us at the University of Washington. Over the last decade, Luis's research has explored computer architecture, processor design, programming languages, operating systems, large-scale parallel computers, security, biology, and almost anything else you can imagine. No wonder he doesn't sleep, he doesn't have time. His work has been prominently featured in the New York Times, Popular Science, MIT Technology Review, the Wall Street Journal, and many others. Please welcome Luis Ceze.
Well, thank you very much, Hank, for the introduction, and especially thank you very much for hiring me 10 years ago. It was a very fortunate mistake, I guess. So as Hank said, I'm a computer architect, so I spend my time dreaming of new ways of building computer systems. And a couple of years ago or so, we got together with Microsoft and formed this new team of very interdisciplinary researchers. So we have molecular biologists, computer architects, coding theorists, fluidics engineers, mechanical engineers, and created the Molecular Information Systems Lab, whose goal is really to really explore how we use molecular biology and use biology in general to build better computers. And in fact, the co-director of the lab on the Microsoft side is here in the audience, Karen Strauss, so you can send all the hard questions to her, please, No, this is not a picture of the team, by the way. So this is a frame of a video from the band OK Go, who's a very creative band. This video has been seen over 50 million times. And we liked this video so much that we decided to encode it in DNA. So when we get this video, this high-definition video encoding DNA, it looks like this. So this faint pink smear here at the bottom of the test tube contains millions of copies of that high-definition video. Okay? Why did we do that? Well, it's not to just be cool. It's super interesting research, but there's a reason, right? If you look at Hank touched at this in the introduction, that we're producing information at an incredible pace. So this green line here is a slightly cartoonish, but based on real data, representation of how much data we are producing. It's growing exponentially, okay? That's the green line. The orange line is the total installed capacity of actual storage devices. Okay? So what do you see here? You see two exponentials with a growing gap. Okay, granted, the green line contains way too many pictures of cats that you probably don't want to store. But the point here is that the proportion of data that you can store is decreasing. So we need to do something about it. Okay? So if we look at the mainstream technology media, like mainstream media for storage, like for example, disk, spinning media, or tape. Tape, it's very much alive and well. I know some of you might remember those, tapes in mainframes. It looked like this, rotating in a big cabinet. Now they look like the size of a deck of cards. They can hold tens of terabytes. Still making progress. Now we have flash storage, solid-state memory made of silicon. This is what's in your thumb drives and in your phones. And then also optical media. They've all been making quite a bit of progress, but they're also quickly approaching their fundamental limits. Okay? But we do think that the ultimate limit of storage is to store data directly in molecular form. Okay, so use as few atoms as possible to store bits. Okay? But when you think of molecular storage, you can't ignore the fact that nature has evolved an incredible information storage molecule. It's called DNA. Right? So life uses DNA to encode information called genes, and genes are essentially blueprints for proteins. And proteins then do everything in your body, right? Their hair, nails, and they move things around your body. They're enzymes. They do a bunch of things to support life. That's what life uses it for. But what we're going to do is we're actually going to store digital data in DNA. So DNA is incredibly general, right? You can store anything you want. And I want to emphasize that we're actually using what we call synthetic DNA. So this is DNA manufactured chemically from scratch. Okay, it's chemically identical to DNA that life used, that nature evolved, except that we're not taking it from organisms, nor putting into organisms. It's really just DNA manufactured and put it in a test tube in vitro. Okay? And in a sense, we are essentially building on the incredible progress that the biotech industry has made in writing and reading DNA. You know, this is a huge industry right now and growing. They made incredible progress. And I like to joke that the computer industry has helped the biotech industry quite a bit in the past. But now that the computer industry is in trouble, the biotech industry has to pay back and help the computer industry, keep storing data. Okay, so let's see now why. So why should we use DNA for data storage? So there's several reasons. First, DNA is extremely dense. Okay, you can store 1 exabyte in a cubic inch. I'm going to put this in perspective in a bit, but this is very dense. Okay, it's thousands and thousands of times denser than the densest thing we can buy today. Okay? DNA is also extremely durable. So we find DNA that are over 100,000 years old, we can still read it perfectly. Okay? Compared to, say, CDs, which last at best a few decades. Okay? And also the other thing that might mean something to people in storage in the audience is that you can copy pretty much arbitrary amount of information in constant time. Okay? In a computer system today, when you drag a file to copy it, it normally takes longer, the file is larger, right? So now if you have data stored in DNA, you can actually copy it essentially in constant time an arbitrary amount of information because it's extremely parallel. We have enzymes that makes copies of DNA for us. So making copies is nearly free. And finally, DNA never gets obsolete. Okay, so as long as there's DNA-based life on Earth, who knows, right? These robots are taking over, right? But it's never going to get obsolete. Unlike the floppy disks that you have in your basement that you probably can't read anymore, right? So this is not going to happen to DNA. DNA is always going to be relevant. So let's put density in perspective. Okay, so this is a picture of a data center that has roughly a few exabytes worth of archival storage. Okay? Code storage, things that you don't access frequently, it's for backups. If you were to put in DNA, it would fit in a few sugar cubes. It's extremely dense. And the reason that this matters is because the cost of maintaining data centers is proportional to the volume of the building, right? And in fact, we're actually we are building a lot of new data centers every month. In fact, probably a new data center being built almost every day. You don't want to have the whole Earth covered with data centers, right? You want to make them smaller. If you remember Colossus, right? It's this giant computer that covered the face of the Earth. We don't want that to happen. We want to make data centers denser and smaller. So let's see where DNA fits in the storage hierarchy. So here I'm showing a typical storage hierarchy up to here, right? So the first 2 layers are storage for things that you access more frequently. We call online storage. At the top, you have flash, which, again, is the type of memory that you have in your thumb drive or in your phone. It's very fast. Its capacity on the order of terabytes. And the durability is of, say, 5 years or so. And that's backed up by hard disk drives. These are spinning media, the ones that make noise. Okay? They're a little bit cheaper. They're bigger, a little bit slower, but still fast enough for online storage. Right underneath that, you have what we call archival storage, or storage for backups. Things that you're going to keep for a long time, or you want to archive it for long periods of time. That's typically stored in tape or optical media. Like Facebook, for example, uses Blu-ray discs to back up your photos on Facebook, and your videos on Facebook are backed up on Blu-ray discs. So the access time is on the order of minutes now, not super fast. Capacity on the order of petabytes, and then durability on the order of decades. So now the way we see DNA is actually fitting right underneath that. with extreme capacity on the order of yottabytes. So yottabytes is 10 to the 24. Okay? Oh, by the way, I should mention this, that people are thinking about what comes after yottabyte. Does anybody know? What is that? Helabyte. A helabyte, okay? So I'm serious. You can look on Wikipedia, okay? So, and of course, because everything in Wikipedia is true, so. Okay, so back to storage. The access time is on the order of hours. And this seems slow, but I want to bring up the point that this is really for code storage. And in fact, there are services today called code storage, that in fact that Amazon offers and Microsoft offers as well, that have service level agreements that are on the order of hours, if not days, to access the data. Because they put in disks and they just stash it away, and then a robot has to come and plug it back in to retrieve the data. So we see DNA right now is really to be the ultimate archival to store all of the world's data in a safe and dense and durable way. Okay? All right, so let's see how this works. This is a mini outline of what I'm going to tell you for the next 15 minutes or so. We're going to start with bits, digital data. We're going to encode into DNA sequences. That's the encoding process. And then we're going to actually manufacture the DNA molecules that are represented by these sequences. That's the synthesis process and put it in the test. So that's the right path. Okay? So let's start with that. So here's the first thing to remember from Biology 101 was that DNA has 4 basic nucleotides, A, C, T, G. Okay? So, and the information in DNA is stored in the order in which you string them together. And the DNA molecule is a polymer made of these 4 basic nucleotides. So you can string them together, kind of like a bead necklace. Okay? You can just put different colors. It's a similar thing. You form a string of DNA nucleotides. So the way we encode digital information, the simplest form is to, since you have 4 letters, is to map 2 bits per letter. For example, we can use 00 maps to A, 01 maps to C, 10 maps to G, and 11 maps to T. Okay, so I can get a long string of bits and map it to a long string of DNA. So far, so good? Okay, except it's not quite that simple for a couple of reasons. Okay? The first reason is that you don't want to have too many repeated letters. So if you have digital data that have lots of zeros or lots of ones that are consecutive, you would have repeated letters. And that's bad because the molecule can be more fragile and it might be harder to read or even potentially more harder to write as well. So we avoid that with clever coding. That's where a lot of computer science kung fu comes into play. We randomize the sequence. We use rotating codes to avoid repeated letters and so on. So we're clever on the computer science side to make this mapping more intelligent. Okay, so the other reason that this is not so simple is that we can't make a very long DNA molecule. So if I have 1 trillion bits, okay, I wouldn't make 100 so billion string of DNA nucleotides. Because when you're manufacturing DNA, there's a probability that you're going to succeed in writing every new letter. Even if this probability is pretty high, say 99% of success, every time you add a new letter, you compound the chance that you're going to fail. Okay, so if you are at, say, 100 letters, you're already at 36.6% chance of actually succeeding in building that molecule. Okay, and 200, you are 13.4%. So how do we fix that? Well, we'll break it up. So we get a long piece of DNA sequence that encodes the data, break it up into pieces, but then now something happened, right? When I go from this to this, I lost the original ordering information that the letters were in the original sequence. So what do we need to do? We need to preserve that information with a label. So we're going to add a little index to each molecule that tells you this is 1 of 2, 1 of 3, 2 of 3, 3, and 3 of 3. Okay, this is analogous to if you have a very large file that spans multiple floppy disks. You have 1 of n, 2 of n, 3 of n. If you ever installed Windows with floppy disks, you have a stack of floppy disks, right? 1 of n, 2 of n. It's a similar thing, except that we get that label in the floppy disk and encode it in the molecule itself. So we know where that piece of information maps back to the original sequence. Okay, the other thing that we need to do is to say that when you have a bunch of molecules that map to a file, I need to say how I group these molecules as belonging to a certain file. So we're going to add tags called primers to the beginning and end of each molecule that says that this molecule belongs to a certain file. So all the molecules of a given file have the same beginning and end tag. Okay? And to put this in perspective again, it's as if each one of these molecules is a tiny little nanoscale floppy disk that can hold 20 bytes. Okay, so a file is going to contain millions and millions and millions of these different molecules. Okay, that when you read, you can piece them back together. Okay? All right, so we went from bits to a large collection of molecules that stores this digital data. So now what we need to do is to think about when we're actually going to write it in DNA, DNA isn't perfect. So we need to deal with errors. When you write and read DNA, you actually have three forms of errors. You have insertions, when you have a letter that wasn't supposed to be there, but it's there because of an error. Or you have deletions, you have a missing letter. Or you just have the wrong letter, that's a substitution. And that happens fairly frequently, about 1% chance per position. Okay? Now, I want to bring up the point, too, that error correction is something that computer scientists do all the time. No computer memories are perfect. So main memory has error correction, disk has error correction, and even communication has a lot of error correction. So we are used to doing that. But with DNA, we have more error modes. Computer memories have only substitutions. With DNA, we have also insertions and deletions, so we have to deal with that. And the way we deal with that is, instead of just storing the payload, we're going to add extra redundant information that are essentially mathematical codes that allows us to retrieve the original information perfectly, despite errors in any of these molecules, and also despite molecules completely missing. Okay? The way you can think about this is that we add enough redundancy here such that if I have any random subset of the molecules that's large enough, I can still recover the file perfectly. Okay, so we extended the codes to support that. Okay, so now we are almost there. We know how to go from bits to a bunch of DNA sequences that contain enough information to deal with errors. Now we need to actually make the molecules themselves. That's the DNA synthesis process. And this is where the biotech industry has been making a lot of progress too. So here's how it works. So there's a surface, okay, where DNA molecules grow. Okay, they kind of grow like grass. So this is the surface. When you look closer, there are little spots that DNA molecules grow on. And each spot here grows a different DNA sequence. And you can make millions of those in parallel. Okay, they grow kind of like grass. Okay, so they keep growing, and when they reach the desired length, in our case, we make molecules of 150 nucleotides. Okay, so they reach 150 nucleotides, we just cut them like grass, put it in a test tube, and there we have our DNA. Okay, so that's an DNA synthesis process. Clear? Okay. Good? All right, so it's amazing. This actually works. These are tiny. A DNA nucleotides on the order of 0.3 nanometers on the side plus, by 0.3 by 0.3 by 1.5 nanometers or so. Okay, and you string them together chemically in a very, very parallel way. Okay, so that's the right path. So we went from digital data all the way to molecules at the bottom of a test tube. But as you can imagine, a write-only memory wouldn't be very useful, right? You need to be able to actually retrieve the information. So let's see how we do that. So we're going to go take the DNA molecule and do what we call sequencing, which is reading the letters encoded in the DNA molecules and then decode that back to digital data. But the first question we need to answer is the following. Suppose that I have a test tube that contains DNA from multiple files. As I told you before, each file maps to a collection of molecules, a large number of molecules. If I put 3 files in it, all the molecules are mixed together. And let's say that I only want the video file here, the one at the top. So how do I do that? Can you have molecules from multiple files? They're all mixed together. There's no molecular tweezer that lets you go one by one and pick the molecules that you want. But luckily, there are enzymes that can do that for us. There are proteins that do amazing things. So here's how we do this. So there's an enzyme called polymerase that can make copies of DNA in a programmable way. Okay, so here's an example. Bear with me. I know it's Thursday night. Don't fall asleep, but this will be worth it, I promise, okay? So suppose that you have 3 files in a test tube. You have a red file, a green file, and a blue file. They all map to a bunch of molecules represented here, and I mix them in a test tube. And let's say that I only want the blue file. So here's what we do. We add a complementary tag, and DNA has another interesting property that it binds to complementary sequences. By complementary, I mean A binds to T and C binds to G. Okay? And if they're perfectly matching, they bind very strongly. Okay? So now, I told you before that all the molecules for a given file have the same beginning and end tag. So if I only want the blue file, what I want what I do is I add this short snippet of DNA that matches the tags at beginning and end. And then I run a reaction called polymerase chain reaction, done by this magic, beautiful enzyme called polymerase that makes copies of DNA in an exponential way. At every cycle, I double the number of molecules that I want. So now if I want only blue, I add the blue tag, I run the polymerase, and then blue crowds out everything else, right? So say if I do 10 cycles, it would be about 1,000 times more frequent than anything else. So when I take a second sample, I only get blue, or mostly blue. So this is how we do random access. And again, nature does this all the time. That's when you go and express certain gene to make proteins, defines the location, copies what you need, and translates this, and so on. We are essentially use that, engineered around that, to extract music videos out of DNA, okay? So, but that's still not sufficient. So what we need to do is now we have a little test tube that contains the molecules that we want. But now we need to know what information is in that molecule. That's where DNA sequencing comes into play. And I know that Seattle has had a big influence in the development of DNA sequencing early on. Then again, DNA sequencing gets the actual physical molecule and tells it and produces a computer string that says what are the letters that were present in that chemical molecule, in that DNA molecule. And that's done by an equipment called DNA sequencer. The one that I'm showing here in the top, on the top is made by Illumina, who is the 10,000-pound gorilla of DNA sequencing today. So they make machines that can read DNA in a very parallel way. So this machine here, we have one in our lab, can read 400 million different molecules in parallel. Okay? And the way they work is by actually flowing DNA on a piece of glass and then flowing complementary letters that glow in the dark. And then they take pictures, and depending on where it glows, they can decode what base was there. Okay? So this machine is normally used for, again, for medical diagnostics, for genomics and life sciences research. We are using to read videos out of DNA. Okay? I promise the last time I'm going to know. Not quite the last time. I'm going to mention this one more time. So the other thing that we are also using in the lab is a new sequencing technology called nanopores, which is really amazing. So first of all, just compare the size. This is the size of a fridge, and this is the size of a little Snickers bar. Okay? So imagine like IBM mainframe, IBM PC. So there's an interesting, duality here, right? So the way this sequencer works is that if you were able to look closer, there's a little grid of holes in the surface that the DNA flows through. And as it flows through, it produces electrical signals that can be decoded into the letters that compose that molecule. Okay, we've actually demonstrated reading digital data with both the de facto standard sequencing as well as this emerging sequencing technology called nanopores. Okay, all right, but that's still not enough, right? Here's what we've done so far. We got the DNA that we want in a test tube. We did the sequencing process, which is going to produce a computer string for every single molecule in that sample. But these strings are DNA strings, not digital data. So we have to do a lot more work. That's where it gets even more interesting from a computer science point of view. The first thing we need to do is to realize that, as I told you, reading DNA is not perfect, has errors. Okay, so how do I actually recover accurate information from a bunch of strings that are all noisy? We do what we call clustering. We compare all of them and form the ones that are similar to each other, form a little cluster in the space. Okay? And in this cluster, I can actually find consensus to determine what was the original information that I intended to put in those molecules. Okay, so I cluster based on similarity. And this is something that if you heard of AI or machine learning, this is machine learning bread and butter. They do clustering all the time. But again, this is applied to reading videos out of DNA. Okay? So we get these noisy strings. We cluster them in space. And with that, I know how to look at the index and put them in the original order. Okay? Once I have that, I can apply error correction to recover the data and then get error-free digital data. Okay, it actually works. We've demonstrated 0-bit errors in quite a bit of data. I'm going to talk more about this in a second. Okay, so here's what we have. We showed you how to go from digital data to DNA sequences to molecules to DNA sequences again into digital data. But I want to talk to you about scalability. Okay, so suppose that I want to store the world's information in DNA. Okay, instead of putting the whole thing into a single test tube, which is not going to work, right? It's going to be, say, a big molecular mass of DNA, right? So it's going to be way too complex. We can't deal with it. So we're going to have an array of test tubes where each little test tube is going to contain a different set of information. This is very similar to a library. If you've ever seen a tape library that has a bunch of tape in a shelf, and then you have a robotic arm that goes and gets the tape and puts it somewhere. It's a similar thing, except that instead of having tapes, we're going to have samples of DNA in a test tube. Except that this test tube is not quite a test tube. It's going to be actually tiny little spots on the surface, about 1 millimeter on the side. So here's a picture that we took in our lab. This spot here, it's actual DNA dehydrated on the surface that's just under 1 millimeter diameter, and it holds about 100 terabytes. Okay? And we're going to have an array of those on the surface packed very closely together. All right, if you had a library of tapes, you have a robotic arm that goes and picks up a tape and puts in the tape head and so on, right? The question here is, how do we do that for these DNA spots, right, these DNA samples on the surface? Well, we're going to build robots, but we're going to build with liquid robots. Okay, so this is a picture of a system that we built in our lab based on a technology called digital microfluidics. Okay, so digital microfluidics allows us to move droplets of water on the surface controlled by software. This is what this video is doing. I love this video. I get hypnotized watching it. So these are droplets of water, okay, that are moving around, controlled by software. Okay, so these are about 1.5 millimeters on the side, so it's definitely magnified here. And they're carrying things, so these droplets can go to a little spot, suspend the DNA, and move it around. So this is our tiny little molecule-moving robot that carries information. And we actually demonstrated that this works. We spotted DNA on glass and had droplets visit that, take a sample, and carry it around. So this is our robot that moves the data in our library. Okay? Great. So let me tell you a little bit about the data that we've encoded so far. As I mentioned before, we encoded this video by the OK Go band. The reason we chose this video, by the way, is because it is about a Rube Goldberg machine. That's super interesting. He moves, it goes, picks one thing, hits the next one, and so on. And in many ways, we see nature as a very, very scalable, as a very reliable, but nanoscale, molecular scale Rube Goldberg machine. Okay, so we use this as a, and then we also included a bunch of other data items like the Universal Declaration of Human Rights in all human languages. We've encoded the Project Gutenberg books, the top 100, the Crop Trust database. That's a super interesting use case because the Crop Trust essentially keeps track of seeds around the world to secure production of food for the future. What we did was to encode their seed database in DNA. So you can actually put together with the seeds the information where the seeds are. Okay? And then the other one is the golden record in the Voyager, which is very timely right now, given all the news around Voyager, right? I think that's the engineered human object that's farthest from the Earth. We got the time capsule and encoded in DNA as well. One day we're going to send it to space, but we'll see when we get there. So the other thing that we did recently that you might have heard about was to encode songs from the Montreux Jazz Archive. So the Montreux Jazz Festival is also associated with this archive, probably one of the largest music archives in the world. And they approached us and our collaborators, Swiss Bioscience and Microsoft, to encode some of their songs in DNA. So we encoded 2 full-quality archival all-tracks songs in DNA as a real use case. And that was interesting to us because, first of all, it worked flawlessly because it was like probably the 20th time we had done it. And also, it's actually a real archive use case. And we see DNA as sort of like the perfect media for archival. It's dense, it's durable, and it's just perfect. So now, in terms of the amount of information that we've encoded so far, we've encoded 400 megabytes of unique data that maps to roughly 5 billion nucleotides. And so we demonstrated random access, and we also demonstrated the ability of moving samples of DNA around with the droplets that I showed you. Okay. So, and this is the largest amount of DNA, of data stored in DNA in the world right now, so by at least an order of magnitude. And we're still growing fast. Okay, you might be wondering, how fast is this today? Well, today it's about 10 megabytes per week. And to put this in perspective, It's just about as fast as an acoustic coupling modem was in the 70s. Do you guys remember those that you you had to dial very fast and then put the headset on top of the acoustic coupling modem? It was about 10 megabytes per week. This is how fast it is today. And in part because we still have FedEx envelopes shuttling around the West Coast carrying data. Okay, when we build our system, we're not going to have FedEx in the loop. Okay, there will be no FedEx. We're going to take this out of the loop to make it a little faster. Okay, so here's where we are at today. We are 10 megabytes per week, but we want to reach 100 gigabytes per second. So there are a few orders of magnitude in there. So you might be wondering that we're crazy. Well, maybe, but I don't think so. So here's some evidence that we don't think we're crazy that we can get there. First, if you look at the trends of progress in DNA writing and reading, it's been incredible. It's remarkable. So in this plot here, I'm showing two things. So first, the transistors on a chip growth, that Moore's law. Moore's law says that you double the number of transistors on a chip, double the density every 18 months. So that's the dotted line. And the green line shows the ability to read DNA, and the orange line shows the ability to write DNA. And they're all growing at exponential rates, in fact, faster than Moore's law. And the other thing I want to point out here is that the life sciences industry has really optimized DNA synthesis and DNA reading today for life sciences applications, which means that the DNA has to be perfect, and reading has to be perfect. If you want to make a medical diagnostics, you want the information to be as perfect as possible, right? And if you're synthesizing DNA for, say, gene therapy or for research, you want the DNA to be perfect. An error there could be catastrophic. But as I said before, in computers, we're used to dealing with errors, so we build error-correcting codes all the time. Okay, so we can deal with very, very imperfect DNA. So that allows us to take trade-offs that the life science industry can't. The other thing that I love if nature had evolved a better error-correcting mechanism for DNA, which means it wouldn't have some issues we have with mutations today. But anyways, it is what it is. Okay, so I talked about storage. So, and let's say if we succeed at storing the world information in a little bit of DNA, what happens to compute? We need to have a commensurate improvement in compute to make use of this information. So I want to transition the talk to some thoughts on how we can improve the compute side by borrowing from nature as well. But before we do that, I want to do a quick thought experiment here. So you keep hearing about AI all the time. It turns out that AI in the end is a bunch of computers crunching very large matrices. They multiply a matrix by a vector, and they do this over and over and over again. That's what AI is, on the compute side. Crunch a bunch of numbers, okay? Now, if you were to build a planetary scale AI system, it would have billions of users. That means billions of edge devices, okay? And many millions of servers to back up those edge devices. That uses easy tens of gigawatts. And to put this in perspective, the largest power plant in the world today is one of the largest in the world. The second one is Itaipu in Brazil, which is 14 gigawatts. And that wouldn't be enough to actually run this infrastructure. So computers use a lot of energy. Okay? And then the other thing that's going on that you might have heard is that Moore's law that I mentioned a couple of slides ago is slowing down, is almost done. Okay, so in fact, even if The Economist announced it, it's because it's true, right? So there was a piece in The Economist last year, Technology Quarterly, that talks about what happens to the IT world as Moore's law slows down or ends. And the response by the IT industry right now has been to specialize the hardware. So if nature has evolved highly specialized computing engines. Your brains have highly specialized computing engine. So Google built this thing called the TPU, which is a processor specialized in machine learning. Microsoft has a reconfigurable hardware in the cloud that's very efficient as well. And that gives you quite a bit of efficiency, but that's not sufficient. Okay, so we still have to do more to actually be able to continue scaling. And one of the thoughts that we've been exploring for the past 5 years or so was to, again, borrow from nature the following idea. So nature does computing all the time, but it does computing that's very approximate. Your brain is doing it right now. You're kind of paying attention, right? But when you really need to pay attention, you can pay attention, then it makes you more tired. In fact, there's this really nice book by Daniel Kahneman, who got a Nobel Prize on models of human cognition. He wrote this book called Thinking Fast and Slow that some of you might have read before. So he talked about system 1 and system 2. System 1 is when you make quick, snap judgments. You think quickly and approximately. That's easier. But if you want to think more detailed, you invoke system 2, which is more precise but uses more energy, and you get more tired. Okay? So we can do the same with computers. If you look at all of the applications today that use a lot of compute power and storage and communication, they don't have to be perfect. There are things like analyzing videos, recognizing voice, and so on. These are things that can tolerate errors in compute and still provide its functionality. We can do the same with computing and do what we call approximate computing, which trades off accuracy for better efficiency, the same way that nature does it. Okay? So we've built approximate compute engines. We built approximate storage for videos and images. If you have a few pixels that are off in an image or a video, you don't really notice it. And so you can relax error correction communications, and you can even relax how coherent the information is when you replicate all over the world. Okay, so this gives you much better efficiency. And to give you an idea, this trade-off can give you between 10 and 100x better energy efficiency. So what we're doing now with nature and the compute, approximating or borrowing ideas from nature and the compute side, is to trade off accuracy for better efficiency. Okay, so I told you a bunch of techniques that you can do in the silicon world that mimics nature in this trade-off to get efficiency, but that's still not enough. And the other thing that I find fascinating about nature is that proteins do everything. In fact, proteins are heavily involved in information processing things in nature. For example, the copying DNA that I showed you, there's an enzyme that copies DNA. So in DNA and proteins is very, very efficient. So a protein uses one ATP molecule as its source of energy. That's about 10 to the minus 20 joules. And if you were to build a computer system that a transistor will be implemented by a protein, so this computer system will be about 10,000 times more energy efficient than a silicon-based computer. That's amazing. So, and that's one of the things that, luckily here in the University of Washington, we have one of the world's best protein designer, David Baker. So we've been talking about ideas on how you can design proteins to actually behave like transistors and build computers out of them. If we succeed at doing that, now we have molecular storage and we have molecular electronics, right, or molecular computing that can be extremely efficient. And with that, I want to close with one thought and how we could build computers in the future. And I think that each technology has its place in building future computer systems. So I spent a bunch of time talking about biomolecules. They can store a massive amount of data, have extreme parallelism, and very efficient. And they also self-assemble. Okay, so to build a silicon chip today, you have to first build a $10 billion fab. Nature self-assembles it. Like really, like, it just finds its way in and self-assembles structures. It's amazing. Proteins self-assemble into incredible structures. So we could use that to build computers too. So biomolecules, I would say, maps to parts of your application that uses lots of data, and it's very efficient. Then I'm sure you've heard of quantum computing. Quantum computing is also amazing. It gives you incredible amount of computation that grows exponentially with the number of qubits. But it's not very good at moving data around. So quantum computing would be great for massively specialized parallel computing, but it uses little data. And then you have electronics. So it's amazing what we've been able to build with electronics. We build chips today that have billions and billions of transistors, and most of them work. And if you look at a picture, they're neatly organized, and they all actually work. Look at a computer. Right? And that could be the perfect control system for computers. And I think we're going to continue engineering with CMOS and electronics for a long, long time, because it's very controllable, very engineerable, and could be the perfect way of gluing everything together. Okay? So with that, I'd like to thank the Molecular Information Systems lab team. So it's a fantastic team of people that really did all of the work that I talked about today.
Let's thank Luis.